Thank you very much for this nice presentation. Thank you very much to be here, to listen to me. It's a very nice hall. Just to be in front of these guys on the photography is quite embarrassing, to be capable to give a talk. You know, I met certainly Professor Klaus Wolf several years ago, and so it's really a pleasure for me. Today I would like to speak about this PPAR, and just to explain why our group is interested in this type of field of the target, and just to report some experiment or some data that we were capable to obtain working on molecules that try to target on this transcription factor. This is a lithography of the old institute. This is San Gallicano when it was built in 1725. And that was considered at the time a quite new approach to provide hospitality to pilgrims coming in Rome just to go to the Vatican, to St. Peter, because it's not far from St. Peter. This old building is in Trastevere, that is quite old neighborhood of Rome, quite famous. And still some activities, they are not related to our institute, but there are still some medical activities in this building, and now we are in this new building that again in the periphery of Rome is close to the ring that in Rome we have, not far from the airport, the ring that we call GRA. And there are two different institutes inside, San Gallicano, which is a dermatological institute, and the Regina Elena, that is the cancer institute. We are the same administration, so both institutes share the same building, and in Italy these sort of institutes are classified institute for research. So basically we are a public institution supported by the Minister of Health to produce some translational research. So we have a research lab, but we don't teach, so we have not a university. And it's a peculiar category of the institution that are qualified a high level of national interest level of institution. So the idea to speak about peroxisome, Proliferator-Activated receptor, is quite interesting target. From a biological point of view, to try to identify the mechanism which is capable to transmit signaling from the membrane of the cells with the interaction with the receptor just to lead the signaling to the nucleus to activate the target gene is absolutely interesting, because most of the new drugs are trying to focus on this situation. And this group of molecules are defining the ligand-activated transcription factor of the nuclear receptor superfamily. So it seems quite complicated. Basically, it's a protein that is activated by specific ligand that is capable to transmit to the nucleus the signaling, activating different type of responses element. That means different group of gene. Basically, nuclear receptor superfamily include receptor for retinoic acid, for vitamin D, for estrogen. So it's quite wide range of different type of receptor, and there is a combination among these receptor. In fact, PPAR is combined with the receptor for retinoic acid to activate the specific the gene, target gene. There are three major isoforms of PPAR, classified alpha, beta, delta, and gamma, that are differentially expressed in the skin and in different other tissues. But basically, these transcription factors are present inside any type of the cell. They control several epidermal aspects related to proliferation, differentiation, control of inflammatory situation, and the barrier repair and the lipid synthesis, with some differences among the three different PPARs and some overlap, because it's not necessarily so clear how to differentiate that. This is related to the fact that the molecules up to now that target PPAR are not necessarily so specific to the different isotype of the different PPAR. So, but certainly you can say that there's some the activation anti-proliferative effect, promotion differentiation, basically amelioration of barrier homeostasis that you can find in different type of PPAR on increased lamellar body formation. And this is just reported from a paper from Professor Schmuth. The capability of the receptor to bind with the ligand probably regulate the type of the gene that are activated by this ligand. And there is also what is defined as post-translational modification of the molecule. So basically, This is quite interesting from a biological point of view and conceptually from a therapeutical or pharmacological point of view. The interaction of this transcription factor, this is something, PPAR is something like an FKB, that's one of the most well-known transcription factor of controlling several inflammatory manifestations. There is a possibility also to modify the protein. So there is a possible, some activities related to the transduction of the signal to the nucleus and some activities that the protein can produce inside the cytoplasm. But basically, this is mostly related to people like me working in the labs, looking for inside the cells. It could be sometimes confusing or not necessarily relevant for the clinicians. But just to take in consideration which is the role that they can have in skin physiopathology and possibly in skin diseases. PPAR gamma specifically, because this is the isotype that is more studied and where we are focused on the interest, control inflammatory situation, the differentiation of different cells, and the proliferation. And correlated to that, they control, we were interested to check for the melanogenesis, the sebogenesis, and the senescence process. We are interested about that. This is me about 35 years ago. I started to work with this group of people at San Gallicano, Professor Nazzaro Porro and Ciro Passi. He is a chemistry, she was a dermatologist. In our group was defined the pharmacological activities of Azelaic acid. I don't know if you know that. Azelaic acid is the Finacea present in the market. Azelaic acid is a dicarboxylic acid that produce several, at that time was defined as inhibitor of tyrosinase and was applied and was indicated to treat hyperpigmentation, melasma. In some cases also lentigo meridiana has been published and treated with this molecule topically. Then Azelaic acid was discovered to be capable to treat acne, and up to now Finacea or Skinuren, the indication is to treat acne, and most recently also for rosacea. So at that time it seems quite confusing, the idea how a molecule is capable to interact with different type of disease, pigmentation, inflammatory situation, acne. That seems to be something not really to put attention about that. It was too generic, not capable to identify the mechanism. So several years after that, we will try to identify which could be the true target for the activity of azelaic acid, and we focus on PPAR. This is because azelaic acid, this is the chemical structure, this is a dicarboxylic acid, a saturated dicarboxylic acid with 9 carbon atoms. It's basically the breakthrough product of peroxidation of oleic acid. No? Oleic acid is what we can find in olive oil. When you have olive oil and you leave the olive oil on the air, it can undergo to peroxidation. When it's a peroxidation, the fatty acid is broken. One of the parts of the fatty acid that is released is dicarboxylic acid and azelaic acid. Was discovered at that time just because Pyrrhospor ovales is capable to produce dicarboxylic acid. And at that time was suggested that the depigmentation in pityriasis versicolor could be related also to the production of azelaic acid. And then was discovered it was capability to have some antioxidants activity, anti-inflammatory activities, and some bactericidal activities. But mostly was related to the capability, what we demonstrated quite recently, that azelaic acid is capable to activate PPAR gamma. This seems to us reasonable just because, as we read at the beginning, PPAR are ligand-related transcription factors. And basically, the major ligand of PPAR are fatty acids and saturated fatty acids or products of fatty acid metabolism. The classical ligand PPAR is prostaglandin PGE2, so products of the metabolism of polyunsaturated fatty acids. So the idea is that after the interaction of an hormone or a factor, growth factors with the cell membranes, there is the activation of phospholipase, the release of some fatty acids from the membrane, and then the possible activation of PPAR. So azelaic acid overlap this indication, so we look at for that, showing that really is capable to activate PPAR gamma and through this mechanism to inhibit inflammatory situation. This can explain why it can work, or it can produce some beneficial effect in the treatment of acne and possibly also in rosacea, because we showed that it's capable to reduce a PUVA-induced senescence of human fibroblasts in vitro. Starting from this point, we will interested in pigmentation. This is the reason why our group is interested in pigmentation, since I started to work with Azelaic acid, to work with inhibitor of tyrosinase, how to treat hyperpigmentary disorders, melasma, and the mechanism correlated to alteration of melanocyte proliferation, and of course also the pigmentation process. So we are focusing on vitiligo, and basically our idea is to try to identify which could be the biochemical alteration of the normal appearing melanocyte in vitiligo patients. This is the approach that we are working. So we suggest that there is some intrinsic metabolic defect in the melanocyte in vitiligo that probably could lead to this increased sensitivity to undergo to a cell death of apoptosis. But from a physiopathological point of view, what it was quite interesting, what we were capable, some people of our group, to demonstrate that normally inside the melanocytes that are stimulated with alpha-MSH, the melanocyte stimulating hormone, or the major hormone capable to induce proliferation in normal human melanocyte, to induce differentiation of this melanocyte, to induce the production of melanin. When it interacts with this specific receptor, the MC1R, that is well known because polymorphism of this receptor are linked with the possibility to develop melanoma, together to activate the classical pathways which lead to the activation of ITF, the major transcription factor leading to the pigmentation, is capable to activate these other pathways, which lead to the release of polyunsaturated fatty acid and to activation of PPAR. This opened a new idea because the people working on pigmentation, working on alpha-MSH, knows that alpha-MSH have a pleiotropic effect as most of the growth factors. Certainly, alpha-MSH is capable to induce pigmentation. It's one of the major factors which lead to have a tanning response following UV radiation. But now we know that alpha-MSH possesses also some anti-inflammatory activities, some antioxidants activities. The response of alpha-MSH is capable to cope against the oxidative stress induced by UV radiation, to induce enzyme capable to repair the DNA damage. And analogous of alpha-MSH, the group of Thomas Luger and Marcus Bohm have patented 4 or 5 different small analogous of alpha-MSH with different possible therapeutic applications, including contact dermatitis. And analogous of alpha-MSH has been tried to be used to treat vitiligo, and it's been done in phase 2 trials in combination with phototherapy to try to repigment patients with vitiligo. So quite wide different capabilities. What we were capable to demonstrate that basically through these pathways, rather than to these classical pathways, alpha-MSH is capable to interact with antioxidants, DNA repair enzyme, and so certainly PPAR ligands are capable to induce pigmentation in skin organ culture. So it's true that this activation can work similar to alpha-MSH. That is capable to promote melanogenesis also in normal human melanocytes, and this is data from our group, and also to activate basically antioxidants, because we have demonstrated that there is a correlation between the capability, the level of antioxidant inside the cells and the amount of the melanin. So basically what we've demonstrated in these couple of papers, that people with pheomelanic or pheomelanin synthesis, together with more peroxidable melanin, they have a lower level of antioxidants. And the combination of these factors leads to a more sensitivity to UV damage. And basically, working with some analogous of PPAR, we were capable to induce antioxidants and also the repair of DNA damage. So some physiological effects of alpha-MSH are related to the activation of PPAR. So looking for PPAR in the pigmentation, we probably will be capable to mimic some of the effect of alpha-MSH. So we are working with some molecules just to induce some tanning phenomenon, to have some more specifically, more physiological photoprotection. That's quite fascinating for the people working on pigmentation to be capable, possibly, we were looking for, to avoid the risk that the specific receptor of alpha-MSH is not capable to work appropriately. And this is just to demonstrate that the activation of PPAR gamma with some specific molecule produce this classified as specific PPAR gamma modulator are capable to activate homeoxygenase, so basically the antioxidant pattern catalase, so to capable to protect the cells against the oxidative stress. So it's fascinating, you know. Frequently the people suggest to provide antioxidants or several types of chemical antioxidants by oral supplementation. It's not necessarily correct, this approach. You know, several years ago we provide 1 gram of vitamin C or 1 gram of vitamin E. Now we know this alteration, the balance of the antioxidants is not necessarily protective, but they can produce some pro-oxidant effect. The possibility to increase the physiological antioxidant will be certainly a more physiological approach to treat some oxidative stress or free radicals generated disorders. But, you know, this is just something working for the lab, some, you know, models, so we can have some relevance in the clinical approach to different diseases. There are several papers looking for the possible use of PPAR ligands even in some dermatological diseases. The most known PPAR ligands are glidazone, pioglidazone, troglidazone, rosiglidazone, chiclidazone. These are drugs normally used to treat diabetes, to control glucose metabolism, and to control lipid metabolism, because certainly PPAR control lipid metabolism. We have seen before that PPAR control the lipid metabolism in the skin, the lamellar body formation, and the epidermal lipid synthesis. So it's been, and also the process related to the modification of the activity of the fibroblast. So there are some indications of the use of pioglitazone in the lichen planus pilaris. There are some clinical effects of pioglitazone and insulin sensitivity on psoriasis vulgaris. So there are some trials in which pioglitazone has been administered to patients with psoriasis with a significant clinical improvement, or in combination with methotrexate also in psoriasis. And there is a quite large pipeline of different molecules targeting on these transcription factors with several different possible indications. And this has provided recently to the idea to have some selective modulators of PPAR. PPAR they have these peculiarities. Since it's quite large receptor transcription factors, what has been demonstrated by chemical groups that the interaction of the receptor with this ligand is capable to modify or is capable to drive the cluster of the genes that are activated by this transcription factor. And this probably is capable to explain why there is this wide range of the different metabolic activities, control of lipid metabolism, glucose metabolism, cell differentiation, cell proliferation, probably because different ligands are capable to induce the activation of different groups of molecules. So several companies are working right now to try to identify ligands specifically acting on anti-inflammatory activities, because probably you can remind that a few years ago, rosiglidazone was considered to be a risky drugs because of possible some side effects, mostly related to some modification of the lipid metabolism and some heart failure. So the idea is to try to have some molecules that are capable to work on just glucose metabolism. There are some, some drugs in phase 1 or 2 to treat diabetes, or just working on lipid metabolism, or just looking for anti-inflammatory molecules. And so we started to work with some molecules that should have some specific target of anti-inflammatory gene. This is just the scheme. The different molecules can interact differently with the receptor or activating different type of gene, so producing some different situation. And this is basically one of the molecules that we are working with to identify possible therapeutical effect in different skin disorders. This is a microarray that we have performed in comparison with this molecule together with troglidazone in keratinocyte culture to show that there is, this is in red all the gene that is overexpressed by the treatment with troglidazone, wherever this molecule was capable to activate a very limited number of genes, and not those related with some specific aspect related to the lipid metabolism. So suggested that really, if we are capable to design something, or we will use some specific ligand of the molecule, we can drive the effect of this transcription factor towards some specific target. So initially, since the idea of PPAR is linked with the anti-inflammatory effect, we checked for the anti-inflammatory effect, working on cultured keratinocytes. We simulated with both TNF-alpha or LPS, and we were capable to demonstrate that either at mRNA level, protein level, the molecule is capable, the modulation of PPAR or the activation of PPAR is capable to inhibit the production of inflammatory cytokines. So we can have some anti-inflammatory activities through different pathways, not just LPS or TNF-alpha, which activate different receptor on keratinocytes. In any case, we were capable to demonstrate the capability to inhibit inflammatory reaction, and this was related with the interaction of NF-κB. Basically, the activation of PPAR produced the stabilization of KB alpha, so inhibit the translocation, the cleavage, the translocation of NF-κ at the nucleus level. This is fluorescence to demonstrate this, and this is by Western blot analysis. But basically, the interaction of NF-κB is capable to inhibit the induction of an inflammatory response. So NF-κB is considered one of the master regulators of the inflammatory response in the skin. To inhibit this can open the idea to use this target to treat several different inflammatory skin diseases. And we observed also that the differentiation process, together with the cytokines, we were capable to demonstrate the inhibition of COX-2, so the production of cyclooxygenase products, prostaglandins, and other peroxides, so peroxidation lipids, and also to improve the alteration of proliferation and the differentiation of keratinocytes produced by AGF, for example. And also, this is just to demonstrate the inhibition of COX-2, and also some alteration of the proliferation process induced by TNF-alpha, and either as hyperproliferation stimulus by TNF-alpha and the alteration of the expression of protein or the keratinocyte differentiation. So basically, in an in vitro model, we were capable to demonstrate that working on this target, working with some specific molecules capable to activate some specific group of gene, we are capable to improve the inflammatory reaction on keratinocytes, to improve alteration of the differentiation process of the keratinocytes, and eventually to inhibit the hyperproliferation stimulus of keratinocytes induced by EGF or IGF. So, possibly interesting because it could be an indication to treat psoriasis. In psoriasis, we can have hyperproliferation of keratinocytes, alteration of the differentiation process, and increased stimulus induced by TNF-alpha or IGF. So possibly it could be some indication. And really, PPAR gamma expression in psoriasis is altered. There is a quite large expression of PPAR, but the localization is at the cytoplasm. There is no, it's missing the capability to have a transport of this transcription factor from the cytoplasm to the nucleus. So probably there is the protein, but it's not capable to work in an appropriate way. This is probably the consequence, or it could be the consequence of some inflammatory reaction, or could be certainly a mechanism which is capable to amplify the inflammatory reaction on the skin. So we tried to use a mouse model of psoriasis. This is the mouse model using IL-21 with a subcutaneous injection. This is based on a paper published in Natural Immunology some years ago from a Roman group from Monteleone and Antonio Costanzo, showing that IL-21 is capable to induce hyperproliferation, alteration of the epidermal structure, and some lymphocyte inflammatory cells infiltrate. Topical application, this is the response after 6 days of treatment. With topical application of this molecule, we were, basically it was an improvement of the quality of the epidermis with a significant reduction of the hyperproliferation, the alteration of the differentiation process, as demonstrated by the analysis of K6 or the involucrin, and also for IL-21. So in an animal model, we were capable to demonstrate that topical application was able to induce an improvement of some features of psoriasis. And then we looked for the capability to control also in lymphocyte with the peripheral blood mononuclear cells stimulated with LPS or TNF-alpha. We were capable to demonstrate that several cytokines produced by the lymphocytes were inhibited by these molecules by the specific activation of PPAR gamma. So quite interesting because you can have some topical application, new possible topical treatment for psoriasis, and possible also some systemic situation. And we observed from skin biopsies that IL-21 and also IL-17 was decreased in vitro treatment. So we started with the phase 1 studies in psoriasis patients, showing that really the molecule is not just the clinical situation because we treated limited number of patients, but seems to be promising. Also because now we know that PPAR are involved in innate immunity, controlling the switch, the activation from the dendritic cells to T lymphocytes, and then the activation of PPAR is capable to induce differentiation of Th2 cells. And now Christopher Schlabach demonstrated also these T9 cells overexpress PPAR gamma, and the stimulation of PPAR gamma induced the differentiation of this subtype of Th2 cells. So there is the capability to interfere with the process of the inflammation, the immune responses. The second point that we checked was related to acne, because PPAR considered related to the pathogenesis of acne. In any case, if the molecule was capable, if the modulation of PPAR is capable to interfere with inflammation, with alteration of proliferation of keratinocytes, acne could be a possible target. Christoph Zuboulis and this group have shown that certainly PPAR gamma are capable to interact with sebogenesis, and linoleic acid as PPAR gamma stimulator is capable to produce this phenomenon. And without going in the details of different experiments, this is a cartoon showing what we were capable to demonstrate with the same molecules, that treating the SZ95, that is sebocyte cell lines, which produce in vitro a pattern of lipids almost equal or similar to what occurs in vivo in human sebum, that the specific stimulation of PPAR gamma is capable to inhibit the hyperproliferation induced by insulin or insulin growth factors, to inhibit the mTORC activation of insulin, so basically to produce an inhibition of sebogenesis and the production of inflammatory mediators, so suggesting that this molecule could act in the treatment of acne. As Professor Smuts told before, our group is interested in this lipidomic assay. We have a quite large metabolomic center with several types of gas chromatography, HPLC, mass spectrometry, and so on. And some people in our group is specifically dedicated to that. And Emanuela Camara, particularly, she published a paper to perform a lipidomic analysis of human sebum, or normal human sebum within acne. And what she was capable to demonstrate that in acne patients there are several modifications of the lipid composition. Basically, it can be summarized with the increase of the acylglycerol in one monounsaturated fatty acid that is called sapienic acid, this monounsaturated fatty acids that is specifically human sebum. So we will try to define how this activation of PPAR is capable to interact with the sebogenesis. PPAR are correlated, the expression is correlated with the differentiation of the sebocytes. So the idea that we have, since the mechanism of induction of the sebogenesis through insulin is not dependent by the activation of PPAR, how a mechanism that activates PPAR is capable to inhibit, to modify the sebogenesis could be related with the process related to the differentiation of the sebocytes. So we just, without going into the details of these different Western blot, what we did, basically we will try to use these cell lines in two different models. In one way, we induce a differentiation with the presence of serum or with high number of the cells, as normally occurs in vivo with the cell lines, that you can induce differentiation where there is a high number of cells or the presence of serum, or without serum to maintain cell less differentiated. And we check this through looking with some marker of differentiation, such as PPAR gamma, some antigens related to the differentiation. What we basically we were capable to demonstrate that the capability of the sebocytes to respond to insulin is dependent to the level. This is data that has not been published. We submitted the paper just 10 days ago. That basically the capability of the sebocyte to respond to insulin is dependent on the level of the differentiation of the sebocytes. In our idea, this is a quite interesting situation. Just to explain, just to skip some of this data, what basically we were capable to demonstrate, we know that sebaceous glands are very small before the puberty. The exposure to androgens produce the proliferation of the sebocyte, the enlargement of the sebaceous gland, and this is correlated with the process of this differentiation of the situation. Right now there is a lot of interest to try to correlate acne with the hyperinsulinemic level and high level of insulin growth factor that normally occur during the puberty because insulin growth factor allows to the increase of the muscular mass. So the hypothesis that we are working on that in the individual with the acne can develop, there is an alteration of the process of the differentiation of the sebocytes, so they miss some step of the differentiation and they remain to be very susceptible to the effect of insulin and insulin growth factors. Just in the last issues of Nature Communication, the UK group working on acne published that individuals with acne, more severe acne, have some polymorphism on gene controlling the differentiation process of the hair follicle and the pilosebaceous units. Just, confirming our hypothesis, or just providing some genetic basis of our hypothesis. So we were capable to demonstrate in vitro that sebocyte with low level of differentiation responds to a very low, so basically physiological level of insulin. So, and they produce a sebum, and this is basically what this graph wants to demonstrate. They produce in vitro a sebum similar to those we were capable to demonstrate in vivo. So we were capable to reproduce an in vitro model to induce an acne-like sebum or an acne-prone sebocytes. That in our idea is quite interesting because it's quite difficult to be defining something like that. And just the activation through this molecule of PPAR gamma, we were capable to induce a differentiation of the sebocyte, to decrease the sensitivity to insulin, to improve the quality of the sebum, so to inhibit the proliferation of sebocyte. So basically activating to at least 3 of the 4 pathogenic events involved in acne, that is sebogenesis, inflammatory situation, and alteration of keratinocyte proliferation, hyperkeratinization. So looking at PPAR gamma, we are capable to modify possibly all the pathogenic mechanisms because probably the microbiome of the P. acnes growth is related to the modification of the sebaceous gland activities. So what we will try to demonstrate is this was true in vivo, or these are just some in vitro model that is a speculation that people working with cells, just combining different mixture, or just to add what we want to see and to check for the endpoint that we want to look. And this is we have done another 2 phase 2 with different concentration. This is the first study that we did with this topical application of this molecule. And what we observed that there is a clinical improvement after 3 months of treatment with the total count of lesions significantly decreased. And with a significant decrease of the inflammatory lesions, but also non-inflammatory lesions. And these are some clinical results that we are capable to achieve with some patients treated with acne. This is another case of acne. This is after 3 weeks, so there is just an improvement after 3 weeks. And this is what we observe after 3 months of treatment. This is quite interesting because this is a quite severe form of acne. At the beginning, this guys was suggested to start with isotretinoin because these are 2 nodules. And it was in 3 months, and then he continues with the longer period, a significant improvement of the manifestation. And this is another more rosaceae acne in not certainly so young lady. And this is another not simple case of acne, showing that really topical application is capable to improve the inflammatory reaction and to improve the clinical activity. But what we did is to try to identify in vivo the same parameters that we observed in vitro. So we collected by a sebu tape, so basically it's a tape applied on the forehead of the individual just to collect the sebum, and with a disquame that is sort of scotch that you can keep some stratum corneum. So we look at for the level of cytokines in the sebum and the cytokines in the stratum corneum. And we look at that following the IL-1, following the treatment, it was a significant decrease of the level of IL-1 both in the stratum corneum and in the sebum, demonstrating that topical application of the molecule is capable to reach the sebaceous gland and to inhibit the inflammatory process in the sebaceous gland. And this is correlated with the decrease of the inflammatory lesions, so probably are capable to precede the clinical improvement. That was also a modification of lipoxygenase, so the peroxidation of the lipids, was significantly modified, and it was also an improvement of the quality of the sebum with the decrease of this specific monounsaturated fatty acid that I told you before. Is called sapienic acid because it's specifically of the Homo sapiens. And this monounsaturated fatty acid is reported to be significantly increased in acne. It could be a sort of marker of the improvement. So we observed that following topical application, we are capable to repeat what we observed in vitro. We reduced the inflammatory lesions, we improved the modification of the lipids, and we decreased the hyperproliferation because we decreased the number of the comedones. So we confirmed in vivo that the in vitro model are really very significant. So we consider the PPAR a quite interesting therapeutical target, probably in different skin manifestations such as acne, psoriasis, maybe other inflammatory skin diseases, and maybe also in pigmentary disorders and alteration of the proliferation process related to with them. And this is basically the dance of the group working in Rome all together focusing on this situation. And thank you very much for your attention.